turn the audio. Okay, so when we left off, we were really on the last slide of this slideshow. So we were talking about the water balance, specifically by regulation of reabsorption. And what we had talked about on Wednesday was mainly how we regulate how much water we're reabsorbing. So we talked about the fact that there is an obligatory water loss, but there's a big range of how much water you can excrete. So our minimum water loss is 440 milliliters, but sometimes you pee a lot more than that in a day. And we talked about how that is really mainly due to the effect of ADH. So antidiuretic hormone is regulating how dilute urine is by regulating the presence of aquaporins in the distal tubule and the collecting duct. So it's an antidiuretic, meaning that if ADH is active, you're not going to have that much water in your urine because it's all going to be resorbed. Not all of it, because we do have this minimum amount we're going to excrete in order to get really toxic kind of solutes out of your body. But we can resorb quite a bit of water. So we do that by activating this pathway. So we have ADH in the plasma. There's a receptor for it on the basolateral membrane of the cells in the collecting duct and the distal part of the distal tubule. It's a G protein coupled receptor, so it activates a G protein like they all do, activating adenylate cyclase, which activates our cyclic AMP pathway, which ultimately basically just makes the cell produce more aquaporin 2 and put them in the apical membrane. That's the whole point of this, really. And this is how we kind of toggle the permeability of that membrane on and off. So if we have these aquaporin 2 channels present, and they're present because we have ADH, then our membranes are going to be permeable to water because we always have aquaporins on the other side of the cell, on the basolateral membrane. So we can resorb water at that point, which we're going to do, as always, because of our osmotic gradients, basically. So these parts of the nephron are going, diving back down into the medulla, which has higher and higher osmolarity as we get deeper and deeper. And basically urine comes out of the deepest part of the medulla. So when we have that membrane permeable to water, water is going to go through it. Okay. So we stepped through the parts of this pathway. And then we talked about the things that regulate the secretion, which are really just osmoreceptors. So they detect osmolarity, and pressure receptors are baroreceptors. So they detect how much volume basically there is in your blood. So whether you need to keep that volume up because you have low blood pressure, which is going to secrete ADH. Okay, so that's where we ended. And the last thing is kind of an addendum to this, is that we're also able to regulate water excretion by changing our glomerular filtration rate. We're usually not going to do it this way. This is usually a byproduct of other pathological circumstances, but it does have an effect on water reabsorption. So usually, and this isn't on your slide set because it's from the renal physiology lecture, but I thought it was helpful to kind of look at this again. So we have the curve here for arterial pressure versus glomerular filtration rate. So we can see that we have this plateau in the center, and that plateau is because we're usually auto-regulating glomerular filtration rate. Sorry, was that a chat? No, it's not. Okay, sorry, I thought that was one of you guys asking a question by texting the chat, but looks like not. Okay, so we have this plateau which means that GFR is normally being auto-regulated. We're keeping it steady because, you know, it's one of our homeostasis kind of variables that we're regulating. And we do that by partially by nervous feedback, but also by tubular glomerular feedback and our mesangial cells. And what was the last one? I'm missing one. Oh, the stretch of the capillary walls in our arterioles. But when our mean arterial pressure is like really low or really high, we're not able to kind of sustain that regulation. So if our blood pressure drops below 80 millimeters or above 180 millimeters of mercury, we are going to see changes to the glomerular filtration rate. And those changes do affect the water excretion rate. So if we have really low blood pressure, so less than 80, then we're going to see a decrease in our glomerular filtration rate. And if we see that decrease in glomerular filtration rate is really just how much plasma is coming out of the blood at the renal capsule, so at the beginning of the nephron. So if we have a low glomerular filtration rate, that means we don't have very much plasma being filtered into our nephron. And the plasma is mostly water, so that means we're filtering less water. Now, if we filter less water and we have less water in that fluid, it's just a sort of logical follow through. If you don't have much water there, you don't excrete that much water from the body because you just don't have the water to excrete. So this is more kind of a just math kind of flow through this system rather than toggling anything about the cells specifically. Same thing if we have an increase in blood pressure. If we have an increase in blood pressure, if it's above 180, it kind of breaks out of our regulation zone. So we have an increase in the water being filtered out of the plasma because there's just like so much hydrostatic pressure, basically. So we remember that that's one of the Starling's forces that affects our glomerular filtration rate, is kind of how hard the blood is pressing on those capillary walls in the glomerulus. So if it's pressing really hard, we're going to end up filtering out a lot of water. And when we filter out a lot of water, if we don't change our reabsorption rate, then we're going to excrete a lot of water, which means that we're going to, lose a bunch of water and get dehydrated in this case. And that's only occurs really in pathological circumstances because your blood pressure should not be that high. But it does affect the water reabsorption in that, like, you still like either conserve or lose water in each of these cases. So you're kind of conserving water if you have low blood pressure, just because you're not filtering that much water through the nephrons. And the reverse, with high blood pressure, you're kind of excreting a lot of water. So kind of getting dehydrated just because of that really high filtration rate. Okay, so these are checkpoint questions, and I have posted answers to these on Blackboard. Okay. So we're going to move, well, does anybody have questions about balance part 1 before we move on to balance part 2? We're going to start talking about a lot more hormones as we move into our second set of balance. Okay. Don't see anything in the chat right now, so I'm going to go ahead and move forward. Okay, so in our second part of talking about balance, what we're really going to be talking about is electrolyte balance, and electrolytes are really just our solutes. So we're going to be talking specifically about sodium and potassium. Calcium is on this lecture objectives slide, but we're not really going to talk much about calcium. So kind of cross off number 4 for yourselves. So as we talk about sodium and potassium, we're going to be talking about our hormones aldosterone and atrial natriuretic peptide, or ANP, because these are going to play a big role in how much sodium we're going to have in our urine versus in our blood, basically. So they're going to be the major stimuli for kind of regulating the sodium balance and therefore our electrolyte balance. And aldosterone is also going to play a role in the balance of potassium. Okay. So we're focusing on sodium balance and potassium balance, and really the bulk of this is focusing on sodium, partially because sodium is important for everything, but also because, as we've been talking about, sodium is one of the big drivers of our water reabsorption, so this is all kind of connected. Okay. So beginning with this, we're going to kind of step back a little and just have some ideas about what sodium does. So we can end up with situations where we have high sodium plasma or low sodium plasma. We have words for those. So we have hypernatremia and hyponatremia. Neither of them is ideal. Like anything, whenever we kind of say it's hyper or hypo, we're indicating that there are problems with that situation because they should be regulated. And this is because sodium is really our primary solute in the extracellular fluid. That's what ECF is. And it's really important for keeping our normal osmotic pressure. So that 300 milliosmoles in the cortex and in our normal extracellular fluid, which is being sensed by our osmoreceptors in the brain, as well as that medullary osmotic gradient that's pulling out water from the loop of Henle and basically keeping our blood at the correct volume. So sodium is the main solute that's doing all of that. It's the most common one. It's also critical to the function of excitable cells. So remember that neurons basically work by letting a bunch of sodium flow in, and same deal for muscle cells. So having the wrong amount of sodium is going to change your membrane potential, kind of, which screws with those systems. So it's really important to be able to regulate the amount of sodium in the plasma because those systems are really important and kind of delicate. Fortunately, we are able to do a lot of things to regulate sodium balance regardless of our intake of sodium. Okay. So we're going to start with the renal tubule, our proximal renal tubule. So we've looked at that before, and we saw that reabsorption in general in the proximal tubules is happening kind of automatically. It's isoosmotic. So we're going to see that sodium is freely filtered at that first part of the nephron. So most of it is going to be reabsorbed in the proximal tubules, but we'll also see some reabsorption in the distal tubules and in the collecting ducts. It's important to note that sodium is not secreted. So we haven't talked about many things that are secreted, but remember that secretion is when we take things from the blood and put them back into the filtrate. So that's not going to happen with sodium. So all of our balancing of sodium content is going to happen by affecting the reabsorption of sodium. Okay. So that reabsorption regulation is going to happen through the action of aldosterone and ANP. So those are the two hormones that we're going to be looking at. It's going to be happening just like when we were talking about the regulation of water balance. This is going to be happening in the distal tubules and the collecting ducts. So kind of that whole first part of our nephron is really not regulating anything. That's just like the normal function of the nephron. And really, whenever we're talking about regulating things like actively with hormones, we're talking about the end of the system, so the distal tubule and the collecting duct. At that point, we're going to have some active reabsorption. And in all of our cells, really, the sodium-potassium pump is going to be driving reabsorption. Okay. So we're starting with the proximal tubule, and we've seen diagrams like this before. So here we see that we have our tubular fluid, so the stuff that got filtered out of the glomerulus is now in our proximal tubule. We see our proximal tubule epithelial cells. So we have our apical membrane, the swirly part. We have our basolateral membrane, this flatter part facing the blood. We have tight junctions between them. And we happen to remember, maybe, that one of the differences between our proximal tubule and our distal tubule is that these junctions are kind of leakier in the proximal tubule, which is part of why the proximal tubule isn't a good place to regulate the reabsorption of things, number one, and also why it ends up reabsorbing more stuff than the later sections of our nephron. Okay, so, and here we see that we also have another barrier to reabsorption, but it's kind of easy to get across. That's our capillary endothelial membrane. It's thin. We got blood on the other side. Specifically, we're thinking about the plasma because that's the water-containing part. That's where our electrolytes are. Okay, so the first part of regulating the sodium balance and also the potassium balance, even though we're not regulating it with hormones here, I'm kind of just talking about like how we're starting that reabsorption because we do have active transport happening even in the proximal tubules. So we're not like regulating in terms of turning on and turning off, but we are putting some energy into this process. So on our basolateral membrane, we have our sodium and potassium pump, just doing active transport of sodium and potassium. It's transporting sodium out into our peritubular fluid. It's transporting potassium into our epithelial cells, into our cytoplasm there. So we have a low sodium concentration inside our epithelial cell because of the action of this transporter. Okay. So when we have this low sodium concentration inside the cell, that means we can use it to move stuff through. And we looked in detail about glucose movement. So in this picture, we can see that we have molecule X being co-transported with sodium into the cell. So sodium is moving down its gradient from an area of higher sodium to an area of lower sodium, and it can like pull stuff with it. So X here could be glucose, or it could be like amino acids or something else. Probably glucose because we like to work with the easiest examples. So that's what's happening there. So that's one of the ways that we're getting sodium reabsorbed because it moves down its gradient into the cell and then would be pumped out actively by the sodium-potassium pump, which means we'd have a high sodium concentration in the peritubular fluid. So that fluid right between the basolateral membrane and the capillary endothelial cell. So if that's high, then we can move to our relatively lower concentration in the plasma. Another thing that we'll see that sodium does, and we'll think more on this when we think about acid-base balance, is that sodium still moving down its gradient into the cell. One of the types of proteins that it moves through is basically sodium hydrogen antiporter. So as we move sodium into the cell, we pump hydrogen ions out of the cell. And we'll see that that'll be kind of important when we're talking about acids and bases. Okay, so we can use that sodium to move stuff into the cell, or we can use that to move stuff out of the epithelial cells. But in both cases, we're raising the amount of sodium here in the cell, pumping it out using this transporter, our sodium-potassium pump, which raises the sodium concentration in the plasma. So that's just our reabsorption. And we also saw that that drives chloride ion reabsorption as well. So the chloride ions basically attracted to those charges moving across the cell, so they travel along with the sodium into the peritubular fluid. So that's this long green arrow that we see. So sodium itself is being reabsorbed, and that drives the reabsorption of a lot of other molecules like glucose and the chloride ions, and it'll be kind of important when we start to think about these protons, these hydrogen ions later. In our distal tubule, which is where we're going to be spending most of today, we see a slightly different thing going on. So we still see that we have our active transport through the sodium-potassium pump at the basolateral membrane. So we can still see this part going on over here. And we can still see that we have that sodium being used to pull other molecules through. So we can see that here we have a sodium chloride transporter. So we didn't see those types of transporters in the proximal tubule, but we have them here. But specifically, what's really different in the distal tubule is that we have channels for sodium. So we can see that actually we have channels for both sodium and potassium in the distal tubule. So one of really the main way that we're going to be regulating the sodium reabsorption in the distal portions of the nephron, so in the distal tubule and in the collecting duct, is we're going to be affecting those sodium channels. And when we talk about potassium, we're also going to think about those potassium channels. So remember, channel, we're going to have those ions moving down their gradients, so from areas of high concentration to areas of low concentration. And we kind of know about the concentration because we can see our sodium-potassium pump, and we know that its job is to keep sodium in the cell low. So sodium can flow into the cell through a channel if we have an open channel. Which brings us to aldosterone. So aldosterone increases sodium reabsorption, and it's going to do that by acting on the cells in the distal tubules and collecting ducts. So it's acting on the principal cells, which are our epithelial cells that are responsible for the reabsorption of water as well as solutes. And we're going to see that aldosterone is a steroid hormone that basically affects these channels on the apical membranes. So it's going to do kind of two things. So the way aldosterone works, oh, no, we don't have some for once. Okay, so the way aldosterone works is that when aldosterone is released and we find it in the plasma, it's a steroid hormone that's actually able to cross cell membranes. So it doesn't need any receptors or channels on the outside. It's going to be able to, by simple diffusion, just diffuse across our capillary endothelium and across the basolateral membrane of our epithelial cell, whether we're in the distal tubule or the collecting duct, and it's going to get into the cytoplasm of our epithelial cell, so that principal cell in the distal tubule or collecting duct. Once there, it does one of two things. Really, it's going to do them at the same time. So it's going to be increasing the number of open sodium channels on the apical membrane. So our sodium is able to flow into these cells when we have open sodium channels. So our aldosterone is going to go ahead and open up those sodium channels, potentially make more of them. And at the same time, if it just did that, if all it did was open sodium channels, then we'd kind of overwhelm our sodium-potassium pumps that are present on the basolateral membrane. If we can let sodium flow in really quickly, then we'd eventually end up with a higher concentration of sodium in the epithelial cell, which would stop movement, right, as we reach equilibrium with the tubular fluid. So aldosterone also increases the number of sodium-potassium pumps on the basolateral membrane. So it's letting more sodium into the cell, and it's also making sure that the inside of that cell doesn't get too concentrated with sodium by actively pumping that sodium into the peritubular fluid. So there is a receptor for aldosterone. So aldosterone doesn't need a receptor on the outside of the cell because it can diffuse across the membrane as the steroid hormone. But there are receptors in the cytosol of the cells to receive aldosterone. So aldosterone is diffusing into the cell, binding to this receptor, and then this receptor, through some mechanism, puts more sodium channels into the apical membrane and increases the number of sodium-potassium pumps that we have in the basolateral membrane. Okay. That's kind of the end of a big, long process, though. So aldosterone doesn't act just on its own. When we were talking about ADH, we saw that ADH was secreted directly by our posterior pituitary, but aldosterone is going to be part of a system. So when we're thinking about like pathways, when we're talking about clotting, we're going to have a pathway kind of like that when we're talking about aldosterone. So we have something called the renin-angiotensin-aldosterone system, which, if you can remember that name, RAAS, already tells you something about the pathway. So that's kind of the order in which we're going to trigger these different molecules. Okay, so the first one is renin, and we're going to talk about renin regulation after we talk about this. But right now we're just focusing on the flow of how we get to aldosterone rather than how we're actually regulating how much aldosterone there is. But we're going to do that by toggling how much renin there is. So I'm just kind of keying you into that now at the beginning of the system, the first time I say the word. Okay, so renin is a molecule that's secreted by our granular cells in the juxtaglomerular apparatus. And I pulled up a picture of the juxtaglomerular apparatus in case you don't remember what that looks like or what it is. But it was one of the ways we were regulating our glomerular filtration rate as well. So what we have happening in our juxtaglomerular apparatus is that we have our afferent arteriole going in, swirling around in this capillary bed called the glomerulus, and then coming out through the efferent arteriole. But our nephron is also squiggly and loopy. So our nephron starts here with Bowman's capsule. Then we have our proximal convoluted tubule, so it twists around. We'd have our loop of Henle, so we'd have our descending limb, our ascending limb. Then we'd have our distal tubule, and then our collecting duct going back down. So there's a point at which the nephron squiggles back, basically past its beginning. So our juxtaglomerular apparatus is that point at which the ascending limb of the nephron loop. So sometimes it's labeled as the ascending limb of the loop of Henle. Sometimes it's labeled as the distal tubule. But somewhere kind of around that junction is going to pass by our arterioles going into the glomerulus. So we have this area where kind of the end of our nephron is passing the blood flow into the glomerulus, which is the beginning of our nephron. So we saw before when we were talking about this last time, way back when we were talking about glomerular filtration rate and how we regulate that, that we have some special types of cells in this area where the end part of the nephron is basically touching the arterioles going into the beginning part of the nephron. So we have these special cells in our distal tubule, or the ascending limb of the nephron loop, called our macula densa cells. And those are the ones we were talking about when we were talking about regulating glomerular filtration rate, because we saw that they can release paracrine factors that constrict our blood vessels. But our blood vessels themselves, so the afferent arteriole, also has a special type of cell in it. So our afferent arteriole that's passing by the macula densa cells in this tubule here have something called granular cells. So these are special cells in that afferent arteriole going to the glomerulus that secrete renin. So that was a long explanation of what renin is, but I find it's helpful to kind of get a picture of where that's coming from, because I know I don't know, time seems really long these days. I feel like it was a long time ago when we talked about the juxtaglomerular apparatus. Okay, so it's secreting renin here basically into the afferent arteriole. So our blood flow through this area containing the nephron, so like next to the nephron, is going to contain renin if we're secreting renin. The capillary walls here also contain angiotensin converting enzyme, or ACE. ACE levels vary depending on what type of tissue we're talking about, and ACE is especially present in the lungs. And the job of ACE is basically to do a conversion on angiotensin. So this is kind of looping back again, but angiotensin is a molecule that's secreted by the liver. So angiotensin is going to be in your blood because the liver is secreting it, so it's just flowing around there in your plasma. Usually it's just flowing around, not really doing anything, until it encounters a renin molecule. So renin is coming from those granular cells in the juxtaglomerular apparatus. And when renin encounters angiotensin, it turns angiotensin into angiotensin 1. Angiotensin 1 doesn't do anything to sodium regulation on its own, but when angiotensin 1 bumps in to ACE, which stands for angiotensin converting enzyme, ACE converts angiotensin 1 into angiotensin 2. Angiotensin 2 is the thing that actually affects aldosterone. So angiotensin 2 is going to give us more aldosterone, which is the thing that actually gives us more sodium, open sodium channels in the apical membrane, and active sodium-potassium pumps in the basolateral membrane, causing more sodium to be reabsorbed. So they're kind of 2 or 3 moving threads here. So we have our angiotensin being secreted by the liver. Oh, actually, I think that's what the next picture is. Okay. So one part of this pathway is that we have our liver. It's secreting angiotensin. Angiotensin is moving through our blood. How do I do this? Sorry guys, I want to see where my pointer is, and I'm trying to figure out how to do that. Okay. Maybe I can't do that. Oh well. Okay, so angiotensin is being secreted by the liver, so we see that over here on the left-hand side. Angiotensin turns into angiotensin 1 if we're secreting renin. Then angiotensin 1, when it hits our capillary endothelial cell, bumps into this molecule ACE, which is an enzyme, turning it into angiotensin 2. Angiotensin 2 causes the secretion of aldosterone, and the specific part of the kidney that's making aldosterone is the adrenal cortex. So the adrenal glands are sitting on top of the kidney, I should say, and then the adrenal cortex is secreting aldosterone, which then kind of dives into the kidney and affects our sodium balance in the kidney, and also continues along in our blood flow through the rest of the body. Okay. So this chart is talking about the direct regulation of aldosterone by angiotensin 2, but it's important to remember that the presence of angiotensin 2 is going to be dependent upon the presence of renin and also the presence of those ACE cells. So in the future, when you talk about various types of drugs you might give people, so there are ACE inhibitors that kind of stop this conversion process. So that's a way that you can treat blood pressure, is by targeting this system, by targeting those angiotensin converting enzymes. Yeah, so there are things we upstream in this pathway that are also going to be important for regulating aldosterone. Okay, but if we're focusing directly on angiotensin 2, it has a couple of actions. So what we're focusing on right now is the effect of aldosterone, the effect of angiotensin 2 on aldosterone secretion. But we can see that there are, I really don't know what that beep is. Okay, so we can see that there are other effects of angiotensin 2 as well. So what we've just been talking about is angiotensin 2. If you have high levels of angiotensin 2, angiotensin 2 basically tells your adrenal cortex to make aldosterone. So high levels of angiotensin 2 will lead to high levels of aldosterone. If you have high levels of aldosterone, aldosterone's job in the kidneys is to increase sodium reabsorption by first pumping a lot of sodium out of the epithelial cells in the distal tubules and the collecting ducts, because it's making sodium potassium pumps, and also it's increasing our sodium channels in the apical membrane so that we can have a quick flow of sodium through those cell walls. This increases our extracellular fluid osmolarity. So we remember that osmolarity is really just telling us kind of about the net concentration of solutes. So it's telling us about the pull on water. So when we have an increase in osmolarity, that's actually something that is going to be sensed by our osmoreceptors. So remember that osmoreceptors are up in the brain. They affect the hypothalamus, and the hypothalamus affects the posterior pituitary. So this increase in sodium due to the effect of aldosterone is also going to affect ADH, which then affects our water reabsorption. So when we increase our sodium reabsorption, we increase our ADH from the posterior pituitary, which then increases the water reabsorption in the late distal tubules and collecting ducts by sort of toggling on and off the permeability of the apical membrane to water through the sort of creation or insertion of those aquaporin 2 molecules in the apical membrane. And this is going to increase plasma volume, so this is going to increase the water in your blood, which can increase your blood pressure. As a really long explanation, but that's kind of the whole flow of this, and this is largely the point of the system. A big point of regulating sodium is really to regulate blood pressure. Like that's why when you have high blood pressure, right, like they tell you not to eat a bunch of salt. Part of that is because the regulation of sodium, which is half of salt, has an effect on your blood pressure. So if we're resorbing a bunch of sodium, we're going to raise blood pressure through this long system, starting with renin, moving to angiotensin 2, which is influencing aldosterone, which is influencing ADH, which is influencing how much water you're reabsorbing. Angiotensin 2 also has 2 other kind of more direct effects that are going to affect blood pressure. One is that it just does vasoconstriction on systemic arterioles. So we didn't talk about it in depth when we were talking about the cardiovascular system, but I think it was like a footnote on one of the slides. So angiotensin 2 directly causes vasoconstriction, so that in itself raises blood pressure, so raises mean arterial pressure. Another thing it does in the brain is that it affects your hypothalamic neurons, specifically making you thirstier. So in addition to causing this sort of water conservation and this water reabsorption, it also makes you want to drink more, which also increases the water in your body. So when you increase the water in your body, you're drinking a lot of water, you're increasing water reabsorption. That all leads to higher blood pressure, which can be a good thing if this is a reaction to low blood pressure, right? So now that we've talked about angiotensin II, which is what is directly creating our aldosterone, we want to think about that, how we get that angiotensin II. And remember that the start of this pathway is renin. So this is our renin-angiotensin-aldosterone system, RAAS. So in order to really start this all up, we need to release renin. And we're going to release renin for a couple of different reasons, some of which have to do with intrinsic factors and some of which are extrinsic factors. So we are going to release renin in response to blood pressure itself, really. So if we have a decrease in pressure in the afferent arteriole, so a decrease in pressure here, for example, going past these granular cells as we're going into the glomerulus, that's something that is going to cause those granular cells to release renin. And so we're going to see decreased pressure in the afferent arteriole when we see decreased pressure in our cardiovascular system generally. So if you have low blood pressure, you're going to release renin. You can also release renin in response to renal sympathetic nervous activity. So that's really the extrinsic factor on this list. So if we're sensing low blood pressure elsewhere, we can increase sympathetic activity, which is going to cause our juxtaglomerular apparatus to release renin. And we're also going to increase the release of renin when we see decreases in sodium and chloride ions in the distal tubule filtrate. So as we have fluid being filtered out of the glomerulus, right? So we have everything that's in the plasma being filtered into our proximal tubule. So our proximal tubule reabsorbs sodium and chloride ions, and then the fluid goes through the loop of Henle, where we see that sodium and chloride ions are two of the things that we're actively transporting out of the ascending limb in order to keep the medulla nice and salty, make that medullary osmotic gradient. So those are two places where we're moving these solutes, which we also call electrolytes, from our system. So under certain conditions, this is going to mean that we'll have drops in sodium and chloride concentration in the fluids that are moving past, like basically past or through this juxtaglomerular complex. So at a certain point, when we see really low amounts of sodium and chloride in the filtrate, which we might see because kind of no matter what, we have to keep that active transport of sodium and chlorine out of the filtrate in order to keep the medulla at a high enough osmolarity to reabsorb water. So sometimes we can see really low concentrations in the tubule filtrate, and under those conditions, we're also going to be stimulating the release of renin. Okay, so this is just a flow chart for renin release. So for each of those different pieces of our puzzle for how we're going to get renin into the bloodstream in order to interact with angiotensin, in order to make angiotensin II ultimately, and then in order to make aldosterone, which is going to affect our sodium balance, and by affecting sodium balance, also potentially affect our water balance. Okay, so they're all kind of related to our blood pressure. So if we see a drop in mean arterial pressure, a drop in mean arterial pressure affects the pressure of the afferent arteriole directly because it's just another part of the cardiovascular system. So that's where we'd see a low drop in our arterial pressure, which means that we'd release renin from the juxtaglomerular cells or granule cells in the afferent arteriole. If we have low blood pressure, we'd also trigger our baroreceptor reflex, which we talked about with the cardiovascular system, but the baroreceptor reflex really just upregulates sympathetic activity. So it upregulates sympathetic activity, and the effect there on the arterioles is going to be an increase in renin release. Low blood pressure also means that we're going to have less blood basically flowing through our glomerulus, which means that we're going to have a low glomerular filtration rate. And if you have a low glomerular filtration rate, so like not a lot of stuff is ending up in the nephron, but you are still actively moving sodium and chloride out of that fluid in order to make your medulla highly osmolar, it means that you're going to end up with quite low sodium and chloride content in the distal tubules. And as that passes past our macula densa cells, which are basically the counterpart of our granule cells, so they're the special cells that are in the tubule versus the granular cells, which are the special cells in the afferent arteriole. So the macula densa cells basically let out like a tiny hormone that we call paracrine secretion. So through paracrine secretion, they're going to affect the juxtaglomerular cells in the afferent arteriole, which are granular cells, which is going to stimulate the release of renin. Okay, so we're going to stop there.